I'll be introducing Dr. Jennifer Orthmann Murphy. Jenn Orthmann Murphy, MD, PhD, is an assistant professor in neurology at the University of Pennsylvania. She completed a combined MD, PhD program and neurology residency at Penn. Then, she pursued a combined postdoctoral and clinical fellowship at Johns Hopkins University. Jenn started her clinical research program at Penn in October 2018 to study the role of glial cells in acquired and inherited myelin disorders. As a neurologist, Jenn cares for patients with multiple sclerosis and leads an undiagnosed white matter disorders clinic. She is the co-director of the H-SPAN Fellowship in Multiple Sclerosis and Neuroinflammatory Disorders. Welcome, Jenn.
Thank you so much for having me. I'm really excited to be here and for this invitation. It's really fun and an interesting challenge to figure out a way to share what we're working on and how to fix the brain. And what I'm primarily interested in is remyelinating the brain. So that's one way that we can fix the brain. And shown here actually is an example of remyelination where each color of these oligodendrocytes represents a different time point in the brain. I'm going to show you much more about that. And so what I hope you're going to learn today, I was going to time myself to make sure I didn't go late. Here we go. Is the basic underlying mechanisms of multiple sclerosis. So I'm a neurologist and I am a neuroscientist. And so I take care of people living with MS, and I have this conversation of how to fix the brain at almost every visit. And I hope that people ask me about research that's going on. And so while what I'm going to tell you is the overly simple version, I hope that you get an idea of the basic complexity and what paths we have going forward to figure out how to make this work. I hope that you end with an appreciation of glial cells from here in Gliadelphia, where there's many of us who are really excited to talk about these cells in the brain, and some of the strategies that are currently being pursued to develop therapy. Okay, so I look at MRIs of human brain all the time, so I want to show you what that is. And so this is a cross-section through the brain where the nose is up top here and the ears are on either side, and this black space in the middle is the fluid-filled area called the ventricles. And the white area is the white matter, and this beautiful cortical ribbon on the outside is the gray matter. So why is white matter white? It's because it's filled with fat, and these amazing complex cells called oligodendrocytes form all these processes and myelin sheaths that interact with axons, and the myelin sheaths are filled with fat. So what happens over the course of development and then over the rest of your life are that neurons, which are these amazing cells that we're sort of ignoring in today's panel, but I'll mention them a little bit, where they need to send signals all over the brain and to the periphery, your arms and legs, how you move, feel, touch, think, smell, sense the world, experience life. Some of those neurons are short, some of them are long, and they need to be efficient and they need to be coordinated. So one of the ways they do that is by having myelin sheaths on some of these neurons where they send out these long processes called axons. And here is an example of two adjacent myelin sheaths. They reorganize the underlying axon and they help that action potential or signal go from one end to the other faster. Oligodendrocytes are also with you for your lifetime. So this relationship is very complex and it's for the long term. And oligodendrocytes interact with not only neurons, but many cells in the environment, participating in metabolic functions that we don't fully understand. And so I did a lot of training to get to where I am. I'm just throwing that in so that everybody can have an idea of what they would want to do when they grow up if they haven't done that already. I did all of my training here at Penn, undergrad, med school, grad school, neurology residency. And then I went over to Hopkins to learn how to take care of people with MS, but also to develop an in vivo imaging platform to look at oligodendrocytes in the surface of the brain. And so that's me at the microscope there, a multiphoton setup. And basically what we do is put a little window over the cortex, literally with a glass cover slip over the brain, and using mice where we can look at green fluorescent protein and these mature oligodendrocytes and where they myelinate axons in the surface of the brain that are parallel to the brain surface and then perpendicular as you dive down deeper from the surface of the brain. And so you can generate pictures like this, where this is a view from the side, where these circles are the cell bodies of oligodendrocytes, and all of these processes are the myelinated axons. So you can see, just exactly like this cartoon, the direction of myelinated axons. And you get more and more oligodendrocytes as you dive down deep from the surface of the brain. And if you look over the top, you can see individual cells and all of their fancy processes that they make. And so if you look at the same spot in the brain over and over and over and over again, then you can follow the fate of individual cells. So these big white circles are individual cells, and all those that the kind of fine process signal are all the myelin sheaths and processes. And most of the cells, once they're there, stick around for weeks. Shown here on the right is this region at baseline, and then 6 weeks later. And you can see that new cells form. And in fact, work that we've done and then work from other labs have shown that you just basically collect oligodendrocytes over the course of your lifetime in the cortex, that you can enhance this with some sort of enrichment protocol, that motor learning is important for forming oligodendrocytes, that playing the violin is important for having more myelin in the white matter. So this is something that you accrue over your life, and obviously has very important roles, because we also know that there's a whole bunch of genetic disorders that primarily affect oligodendrocytes, and if you screw up myelin, many things are screwed up. But the poster child of white matter disorders is multiple sclerosis, which is an acquired disorder. And the shortest summary I can think of is that it's an immune-mediated disorder that leads to damage to myelin, or the term demyelination. And the National MS Society has a wonderful description of this disorder on their website if you want to read more. But here's some important points I want to mention. Nearly 1 million people in the US have this diagnosis, so it is not rare. The signs and symptoms of MS are variable. It basically depends on where the lesion shows up. The symptoms can get better, and sometimes they don't get better, and that's progression. And so sort of a typical presentation that we often see is that someone develops over a couple of days loss of vision in the eye, and it hurts to move their eye around, and we find inflammation in their optic nerve. But what is the cause of this in the first place? So we don't know. It seems to be a combination of things. It can be due to inherited risk factors. It could be environmental factors. Sometimes you don't have enough vitamin D or exposure to the sun. And there's definitely immune dysregulation. And how that manifests in any individual person is hard to predict. However, we have 23 FDA-approved disease-modifying medications that all target the immune system. So we can diagnose and follow MS using MRI. So this is the version of the MRI where now the white matter looks gray and the gray matter looks white. But that's because when there's lesions, they're highlighted and look brighter. So you can see all these little ovoid lesions. They're basically extending along venules out from the lateral ventricle. And we use those when they're typical shape like this is in location. Then if there's evidence of active inflammation or breakdown of blood-brain barrier, like shown here, where there's this open ring, excuse me, then we have basically what's dissemination in space and time. There's multiple lesions, which is the name of MS, and evidence of active inflammation. And we can follow MRIs over time to look for onset or lack of onset of new lesions, which is our goal. And we look in white matter. And our treatment goal is to have no more lesions. Despite the ability of having so many really fantastic medications that basically involve an hour talk with people with a new diagnosis to come up with a plan for what to do, because we have so many, there still is progression. One of the ways that we can measure that is shown here with this expanded disability scale. And this particular one looks at motor function and the ability to move around or walk, eventually needing a cane or a wheelchair. Where does the progressive part come from? Well, to explain that, I'm going to give you my overly simplified version of MS. And so on the right here, on the left side here, we're inside a blood vessel where I'm highlighting the types of white blood cells that can be circulating through. Now, you've already heard about macrophages from Chris. I'm not going to really talk about them more because B and T cells, which are lymphocytes, are the main mediators, although macrophages are also important. And as you already heard, there is a blood-brain barrier that separates what's in a blood vessel with the brain. Now, that doesn't mean that cells like B and T cells and macrophages can't go in and check things out and then leave again, and they do that. But they don't do that on a large scale, and they usually don't stay for very long. So in the brain, on the right side, you have the neuron. I've already introduced you to the neuron sending signals all over the place to make your brain function. The oligodendrocyte, which helps make it more efficient and contributes to metabolic support and communication with other cell types in the brain. An oligodendrocyte precursor, which is the most proliferative cell in the adult brain and can differentiate or turn into an oligodendrocyte if need be, but also has its own set of functions and persists over the course of life. Excuse me. There's also astrocytes, which I'm depicting as a little cloud here because they form so many little tiny processes that interact with so much of the neuropill that it's actually hard to schematize this, but they know everything that's going on. They are the busybodies. They're talking with the neurons, they're talking with the axons, they're talking with the oligos, the OPCs, the microglia. And this little process here is basically a process that interacts with the blood vessels. So astrocytes know everything that's going on. And then last, but not actually the last cell, but all I'm going to talk about today are microglia, which you already heard about, the brain resident macrophages. So what happens when the blood-brain barrier breaks down? And whatever type of immune cell, typically the lymphocytes go in and cause destruction, either antibody-mediated or sending out factors that cause destruction. Well, in the case of MS, oligodendrocytes seem to be a main victim in their loss. And what are the consequences of that? Astrocytes become reactive. So they're typically shown as star-like. I just made them look a little bit angrier here. Microglia will phagocytose and clean up things. So I made them into little Pac-Men. And sometimes the axons degenerate and neurons die. Often in lesions, there's fewer precursor cells present. And so eventually the lesion will be left as a scar, and it never goes away, but you're left with a scar. And it's not really clear what role the remaining cells are playing. The OPCs that are there can form new oligodendrocytes and remyelinate, and they seem to have features that are different than the oligodendrocytes that were lost. But the current available disease-modifying strategies are really targeting the immune cell role. The really effective ones just block those B and T cells from getting in there in the first place. Some of the strategies just target B cells alone, and some of them target both B and T cells, either killing some of them, all of them, or hiding them in lymph nodes. There's also additional ones that I'm not going to get into today, but you can see that really none of them address the damage that's left in the brain. Another thing I want to mention is that in cortical lesions, or that ribbon that I showed you on the MRI that's on the outside of the brain, B, T cells, and macrophages actually seem to accumulate there and then don't go away. And that happens over the course of MS, even early on. And it's not really clear what the consequences of that leptomeningeal inflammation is and what to do about it. So I didn't actually tell you that there's cortical demyelination in MS, and it's not visible on clinical MRI. We can see white matter lesions and we can't see gray matter lesions. We actually only know from pathology. Shown here is this brown stain represents myelin, and this is the surface of the brain. And this is an example of a demyelinating lesion, but we don't know how it related to any phenotype or symptoms or cognitive difficulties. And so how do we repair a lesion when it shows up? And so strategies for repair include making new oligodendrocytes, protecting those neurons so they don't die, targeting microglia and astrocytes in the lesion, and stopping any persistent inflammation. And so to make a new oligodendrocyte, you need to get the oligo precursor cell to make a new oligodendrocyte. And the ways that you can do that are to increase the pool, make them proliferate and turn over, have them migrate into the lesion, or transplant them in, sort of like how Chris described for microglia. Or you make more oligodendrocytes by differentiating the OPCs. One of the things that we learned recently is that OPCs actually act as immune cells in MS lesions. Shown here are two examples of those reports. So actually, in addition to these classic strategies that we thought would work, we actually might need to remove or modify these disease-associated OPCs. There are screens that are developed to look for small molecule drugs that can promote differentiation into oligodendrocytes. This is a really cool nanofiber assay from Jonah Chan's lab. This compound, clemastine, actually went to clinical trial. And the outcome of this was that it was safe and there was faster visual pathway signal propagation, which is what we think myelin should do, but we don't really know when to give these medications and how will we know it works. So as a postdoc, I developed a platform to figure out how we could study specifically cortical oligodendrocyte loss and replacement along with the role of all those other cells, which is something we don't really know much about in people with MS. And so to do that, I used the same platform I showed you before, which is look at oligodendrocytes on the surface of the brain. And then in the model, oligodendrocytes die due to exposure to cuprizone, which is a toxin-induced model. And then after you stop cuprizone, new oligodendrocytes are formed for remyelination. And so now I'm going to show you remyelination. So this is an example of the baseline region of cortex. So this is your cohort of oligodendrocytes. And I'm going to show you this region weekly image. And I just want to highlight a few things. So the pink arrow is an oligodendrocyte which will be lost. The green arrow is one that will appear. And the blue arrow is one that is stable over the course of the experiment. And so you can see over a course of cuprizone, oligodendrocytes disappear, new cells appear, and they're so bright in their signal. And that one cell is over here is a survivor of the experiment. I'll show you again so you can take a look at all the awesome things that are happening. All right. So one of the things that you might not have noticed is that when you lose a cell, so here's an oligodendrocyte at baseline, by 3 weeks, almost gone. By recovery of 2 weeks, it's gone. And then a new cell appears in a new place. So there's a new pattern of myelin forming. The other thing that's really important to notice is that as you go down from the surface of the brain, that not all of the oligodendrocytes are replaced. In fact, if you sort of quantify every single oligodendrocyte in that region of brain, only about 50% in that region are replaced. And so we have a platform to study the loss and replacements of oligodendrocytes in the cortex, but repair is incomplete. And so what we're working on in my lab is targeting microglia and astrocytes in the lesion to figure out if they're the problem that's happening in those deeper layers. And so what we found actually, even at 5 weeks of recovery, that we had these nasty morphology in astrocytes that persisted even through recovery. And so Joseph Gallegos, a graduate student in my lab, is going to work on defining how astrocytes change function over the course of damage and recovery. And you can do that by looking at them simultaneously with oligodendrocytes. And Hannah Liu, another graduate student in the lab, is going to look at what microglia are doing. This is a picture of a brain treated with cuprizone, and this is the surface of the brain. And down deep, those microglia have a different shape. And if you look closely, they're actually depicting as a Pac-Man. They are actually chewing up myelin here. And so we also need to define how microglia are changing their function over the course of damage and recovery. How is this interfering with recovery? And then the last thing that I'm going to tell you about today is that we're working on developing a model to better understand the mechanisms of persistent inflammation. It's actually really hard to model this. And so one of the things that we're developing is a model to introduce these T and B cells in a way that we can visually see them. So here's a red T cell dancing around the dura over the brain that hung around there and moved around for weeks and weeks and weeks after introducing it to the mouse. And then experimental autoimmune encephalomyelitis is this classically used model of MS, although it doesn't typically affect the cortex. But if we're able to tweak it correctly to do this experiment, you can see that the T cell will interact directly with myelin. And so I'm going to stop here, introduce the rest of my lab. This is us hanging out at the biopond, which is beautiful right now. And thank my current rotators, as well as acknowledge the Center for Neuroinflammation and Experimental Therapeutics, led by Amit Barawar, and the rest of our MS division, as well as my collaborators. The Burgos lab, where a lot of this platform was developed, including Cody on an oligo right here, funding sources, and our Gliadelphia efforts. And I can't not mention a few things that I tell all my patients for going forward and keeping healthy. And these are actually modifiable risk factors for MS. as well as preventing inflammation, which are eat a heart-healthy diet, exercise every day, even 10 minutes. Just come up with something. Consider getting vaccinated against COVID-19 and ask someone about it if you're not sure. Mask up and ask for help. There's so much stuff going on right now, and it's okay to ask for help. Thank you.
Thank you, Jen. Great talk and beautiful animations. So I have the first upvoted question is from Jay, and they ask, is there any thought or evidence of oligodendrocytes in the brain/white matter different from those that ensheath the optic nerves, which are, of course, outside of the brain anatomically?
So the optic nerve is still within the brain, so they're still oligodendrocytes. Now, the oligodendrocytes are probably pretty heterogeneous in different areas of the brain, different areas of the white matter versus gray matter. They have different shapes. They may not actually form the exact same number of sheaths, although it's probably very similar. We don't actually know yet without being able to follow individual cells in all the regions.
I'm going to ask you a follow-up from that, just to take over as a moderator. Has anyone looked at differences transcriptionally or based on protein expression of different oligodendrocytes in different regions of the brain?
Oh, sure. This is basically ongoing work and things that we're interested in, many labs are interested in looking at. I think one of the coolest things about this is when we find new markers of oligodendrocytes that basically over the course of their becoming an OPC to a mature cell, they express different markers. When they're mature, even within the same region, there's different cells expressing different amounts of different markers. And they form sort of from the back of the brain and spinal cord forward, especially during development. And what tells them to turn on at which time? These are all questions that we don't really know.
Great. Such an active field.
Sorry, one more. Sorry, I don't have to add more, but I will anyway. As a graduate student at NGG, I studied just the gap junction, so the connections that oligodendrocytes make directly with other cells. And even in different brain regions, they express different patterns of oligodendrocytes. And that's just one way in a mature cell. So that doesn't even get to the complexity of everything.
Great. Thank you. Okay, here we go. So the next question is again from Jay. So in multiple sclerosis, even though the main problem is demyelination, after injury to the oligodendrocyte, are the underlying neurons also injured? Do you believe that if you deplete the oligodendrocytes for remyelination, the neurons themselves are able to recover? So
this is a great question, and I kind of just totally ignored the neurons in this talk. So not all neurons are lost. Not all neurons that are demyelinated are lost. They seem to be sick. Some of them get replaced. We don't know the time frame of how quickly we need to repair them. In the particular model that I'm using, there's some evidence that axons get sick, but they're not lost. And so what's really important to know is how fast we need to remyelinate them. What are the long-term consequences of reorganizing an axon so that myelin can be there? And then what happens when you lose that, the contribution that myelin is making to that axon. But in the end, we know that there is loss of some axons and there is brain atrophy over time. And so we have to do better to fix the brain to prevent that from happening.
Yeah, thank you. Okay, this next question is from Ann Matthews. I'm not great at pronunciation, so if I mispronounce this drug, I'm so sorry. Is azilimumab research moving forward successfully?
Sorry, is
elezanumab, ELEZANUMAB.
I don't know.
I'm unsure too. Okay.
I'll have to look that one up.
Yeah, that's one to look up. Okay. I think there was one more here. Jen, this is from Conrad. Is the oligodendrocyte growth enhancement caused, wait, I have someone in the background who's highlighting my slides for me. Is oligodendrocyte growth enhancement caused, does it cause motor learning specific to motor regions?
Well, I'll answer the question the way that I want to. So all of the stuff that I showed you was in the somatosensory cortex. but Ethan Hughes, who's also an NGG graduate, and I worked with at the Burgos lab for my postdoc, and now has his own lab in Colorado, looked at the motor cortex particular, also with demyelination and remyelination, and found that motor learning actually promoted remyelination. That sort of fits in with the idea that we can intervene, we can help reprogram maybe those OPCs to make the cells rather than doing whatever other function they're doing, but we still need to figure out exactly how to do that and what's standing in the way.
Great, thank you so much. We'll see you back for the speaker panel.
Sure.
We'll move forward. Thank you.